Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Biomodels, LLC, I'd like to welcome you to Animal Modeling of Disease States with a Microbiome Component. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now, I'd like to introduce our presenters for today. First is Caitlin Perello, PhD, scientist with Biomodels, LLC, and our second presenter is Benjamin Cuffo, PhD, principal scientist with Biomodels. Welcome, Caitlin and Benjamin. The presenter ball is yours.
Thank you, Elizabeth, for the introduction. Ben and I would like to start by thanking everyone for taking the time to listen to our webinar entitled Animal Modeling of Disease States with a Microbiome Component. Before getting going, we wanted to start with a little bit of background on Biomodels for those of you not familiar with our organization. Biomodels is a preclinical contract research organization located in Watertown, Massachusetts, and we were founded in 1997 out of the Brigham and Women's Hospital. We specialize in highly translational models of human diseases and conditions for a wide variety of clients, with therapeutic expertise in a number of disease states, including inflammation and autoimmunity, microbiome studies, oncology, metabolic disorders, and cancer supportive care. Biomodels has facilitated over 50 compounds into patients for multiple indications. Just to go over our objectives for today, we'll introduce the role of the microbiome in various human disease states, discuss the experimental approaches that can be utilized for probing disease models with a microbiome component, and describe the importance of the microbiome when modeling diseases in vivo. A brief outline of how we've structured our talk today. We'll begin with an introduction, move on to strategies for assessing microbiome effects in disease models, including manipulation, maintenance, and assessment. This will be followed by examples of how the microbiome influences disease modeling, and then we'll finish up with summary and conclusions. The role of the gut microbiome in human health is certainly a popular topic in the news. All of these headlines, sourced from publications such as the New York Times, the Washington Post, and NPR, are from articles published within the last year and a half. And the microbiome has indeed been purported to play roles in a myriad of human conditions, including conditions such as mood disorders and autoimmune disease. Though the bulk of this talk will focus on the gut microbiome, it's important to keep in mind that we have many microbiomes. Bacteria colonize almost every environmentally exposed surface of the human body. And though the greatest quantity and diversity is found in the lower GI tract, biomes exist for the skin and hair, as well as other mucosal surfaces like the mouth and nostril. The gut microbiome refers to the trillions of bacteria that colonize the human intestinal tract, and these include thousands of species-level phylogenetic types. These bacteria come in a couple of different flavors. Commensals, also known as mutualists, are permanent resident bacteria that provide no benefit or detriment to the host, at least not to our knowledge. Symbionts are health-promoting members of the gut microbiome. They live in homeostasis with the intestinal immune system and play roles in nutrition and metabolism, as well as in the prevention of inflammation. The third kind are pathobionts. These are permanent residents of the microbiota that have the potential to induce pathology. When in homeostasis, these bacteria are balanced and don't cause disease. This is referred to as dysbiosis. If there is an unnatural shift in the composition of the microbiota, whereby either the number of symbionts are reduced or the number of pathobionts are increased, the bacteria can become unbalanced. The result is called dysbiosis, and dysbiosis is associated with multiple disease states, including inflammatory diseases like inflammatory bowel disease, autoimmune diseases like rheumatoid arthritis, allergic diseases such as asthma, and metabolic disorders like obesity. In addition to its role in human health and disease, several preclinical studies support a role for the gut microbiome in the therapeutic response to anti-cancer drugs, including traditional chemotherapeutic agents like cyclophosphamide and cancer immunotherapy compounds like anti-PD-L1. Ben is going to expand on this role in particular a little later in the webinar. Genetics plays a role in the development of dysbiosis to some extent, as mutations in NOD2 and the IL-23 receptor are associated with dysbiosis. However, much of dysbiosis is likely caused by environmental exposure and circumstances and events including route of birth, lifestyle, and medical disease. Given the gut microbiome's many roles in human health and disease, animal models that allow us to study these roles are important tools. However, the microbiome is a significant consideration in animal modeling, even when modeling diseases without a strong microbiome component, as evidence suggests that the intestinal microbiome can impact animal model disease penetrance and severity, therapeutic efficacy, and reproducibility. So that's actually a really great transition to the next part of our webinar, strategies for assessing microbiome effects in disease models. Here we're going to discuss microbiome manipulation and maintenance, as well as some assessments. The first strategy we're going to discuss is also the most straightforward, vendor comparisons. This strategy allows us to ask the question, do different microbiomes yield different results? Data from the literature suggests that there could be substantial differences in the gut microbiome composition between animals of the same strain that are sourced from different vendors. While there may also be some genetic differences in mice from different vendors, we can take advantage of the microbiome differences and compare disease development and therapeutic efficacy. Differences in these parameters, comparing mice of the same strain sourced from different vendors, supports a role for the microbiome. If this result is observed, follow-up experiments involving co-housing of and fecal microbial transplants between the mice from different vendors can more directly discriminate between microbiomes versus genetic effects on the model. A pro of this model is its cost-effectiveness. However, the mechanistic data generated can be limited. The second manipulation strategy that I'm going to discuss allows us to assess how direct manipulation of the intestinal microbiome changes disease phenotype. The manipulation can be carried out by administration of antibiotics or by dosing animals with either various bacterial compositions or fecal microbial transplants, or both. Fecal microbial transplants, also known as FMTs, can be sourced from different mice, whether naive or diseased, or from human donors. These human donors can in turn be healthy or may have a disease of interest. Antibiotic administration prior to or during the disease model addresses how gut microbiome manipulation changes the disease phenotype. It can also be used to deplete the gut microbiome before dosing animals with novel bacterial compositions, or FMTs. Since these experiments are performed in conventional animals, which already have a full microbiome, complete engraftment with a novel microbial composition can be a challenge. Antibiotic administration can act to pre-deplete the microbiome ahead of dosing. Alternatively, daily dosing with high CFU can act to overwhelm the system. This strategy is also cost-effective, and since different compositions can be tested, it can provide some mechanistic data. However, since animals have a full microbiome upon dosing, the microbial compositions with which the animals are dosed are not the only microbes living in their gut, which can complicate interpretation. The third strategy is the use of germ-free mice. Germ-free mice completely lack an endogenous microbiome and are therefore the most clear-cut way to establish whether the microbiome is necessary for disease development or therapeutic efficacy. These mice can also be reconstituted with novel bacteria compositions, or FMTs. Since the bacteria with which the mice are dosed are the only bacteria present in the animal, this system can be used to cleanly explore phenotype differences based on different microbial compositions, and to also test bugs as drugs. The lack of a competing microbiome makes these studies very mechanistic, and the animals do not require antibiotic pre-treatment for recolonization. However, the husbandry practices needed to maintain germ-free status, as we'll be discussing shortly, are resource-intensive. So once we've manipulated the microbiome, how do we maintain that manipulation? We do this through the use of various biological barriers. Biological barriers are designed to keep lab animals free from contact with microbes. Each level of barrier is defined by the microbes that are excluded and by the methods needed to create and preserve that particular barrier. I'm going to start by discussing Biomodels' conventional barrier. Animals for experiments performed with conventional housing are sourced from a given vendor's mouse pathogen-free barrier level. Upon arrival at Biomodels, animals are housed in conventional HEPA-filtered cages and are maintained on irradiated food, bedding, and enrichment. All animal manipulation required for the study, such as disease induction and dosing, is performed on the benchtop. Several strategies for assessing the role of the microbiome can be utilized when animals are housed conventionally, including vendor differences, and daily dosing with high CFU with or without antibiotic pre-treatment. The pros for conventional housing are its cost-effectiveness and that there are few restrictions on animal strain availability at the mouse pathogen-free vendor barrier level. However, as we've discussed, these mice have a full microbiome, making recolonization difficult. Furthermore, as animals are manipulated on the benchtop, they are exposed to microbes beyond those with which they are dosed. The next barrier level is specific pathogen-free barrier. At Biomodels, we refer to this as our SPS++ housing. Animals are sourced from the vendor mouse pathogen-free barrier level, as they are for conventional housing. However, upon arrival at Biomodels, mice are housed in HEPA-filtered cages in Biomodels' dedicated SPS++ vivarium and are maintained on autoclaved food and water, bedding, and enrichment. All manipulation throughout the study occurs in a biosafety cabinet. Vendor differences and daily dosing with high CFU with or without antibiotic pre-treatment can be performed at this barrier level as well. But a major pro of this barrier level is the decreased contamination potential, since the animal's contact with environmental microbes is limited. However, as with the conventional housing level, the animals have a full microbiome. The final barrier level I'm going to discuss is the germ-free or gnotobiotic level. As we've discussed, germ-free mice lack a microbiome, as they are born and raised under germ-free conditions and germ-free isolators. Once recolonized, they are referred to as gnotobiotic, but are still housed under germ-free conditions and germ-free isolators, minimizing environmental microbial exposure. Among the first germ-free animals were those derived and reared by James Rainier in 1939 at the laboratories of bacteriology at University of Notre Dame. These animals were derived by C-section and were hand-raised in isolators. Today, mice that are bred in the germ-free environment are the primary animals used for germ-free work. At Biomodels, we source germ-free mice from the Taconic germ-free facility. Upon arrival at Biomodels, animals are housed in Biomodels' germ-free isolators and are maintained on autoclaved food and water, bedding, and enrichment. The animals live in the germ-free isolator throughout the experiment, and all manipulation, including disease induction, body weight assessment, and dosing, occurs within the germ-free isolators. Biomodels currently has 14 germ-free isolators, each of which can house between 20 and 40 mice per isolator, depending on how the animals are caged. There are a lot of pros to the germ-free and gnotobiotic housing level. The lack of a competing endogenous microbiome not only makes the animals easier to recolonize, but also removes that variable if trying to trace phenotypic differences back to a particular microbial composition. Furthermore, as these mice are maintained under strict aseptic conditions, the contamination potential is substantially decreased. All items necessary for the study, including housing materials and disease induction materials, are sterilized by steam sterilization by autoclaving, or by cold sterilization with a chlorine dioxide-based sterilant, or both. And we monitor the germ-free status of germ-free animals throughout the study by culture or by PCR. However, a limitation of this housing level is the limited number of germ-free mouse strains that are available commercially. I'd now like to discuss some of the strategies Biomodels utilizes to assess phenotypes in microbiome-based studies. Both aerobic and anaerobic bacteria culture can be performed in Biomodels' microbiology facility. We can assess intestinal colonization dynamics during the study by the collection and culture of feces at defined time points during the study. These samples can be cultured for vegetative and spore enumeration under aerobic conditions in a biosafety cabinet or under strict anaerobic conditions in a void chamber. In addition, Biomodels can culture and prepare bacterial compositions for dosing under either aerobic or anaerobic conditions. One of the hypotheses for how the gut microbiome plays roles in disease states is through its interactions with the immune system. Evidence suggests that certain bacteria species can have effects on T-cell subsets present in the intestinal lamina propria. Biomodels can assess the lamina propria T-cell subsets in mice dosed with various bacterial compositions by isolation of lamina propria lymphocytes. This prep yields a clean population of CD45 positive phagocytes, as well as CD4 and FOXP3 positive T cells by flow cytometry. So just to briefly summarize this section, we've discussed several strategies with which to assess potential roles of the microbiome in disease modeling. These have included manipulation and maintenance of the microbiome, whether by comparing disease phenotypes of animals sourced from different vendors, modulating the microbiome by antibiotic pre-treatment or dosing with bacterial compositions or FMTs, or using germ-free mice, as well as the different housing barrier levels that are offered by biomodels. We've also discussed the ways that biomodels can assess microbiome-based phenotypes, such as fecal collection and culture, as well as when appropriate lymphocyte prep and analysis. So with that, I'm now going to move on to discussing some examples of how the microbiome influences disease modeling, and we'll go over a couple of different disease states and models. As we've discussed, the gut microbiome has been demonstrated to play roles in various human disease states, including rheumatoid arthritis, inflammatory bowel disease, and obesity. It is hypothesized that this role is mediated by the microbiome's interactions with the immune system and our diet. I'm going to start with a study assessing the role of the microbiome in a novel rheumatoid arthritis model. But first we'll go over some RA background. Rheumatoid arthritis, or RA, is an autoimmune disorder that affects tens of millions of people worldwide. It is characterized by joint inflammation and is of an unknown etiology. Biomodel's standard RA model is the type 2 collagen-induced arthritis model, in which animals are immunized with type 2 collagen and arthritis and pus swelling is observed. As the literature supports the role for the microbiome in RA, both in the clinic and in disease modeling, we chose to compare animals sourced from different vendors, Jackson Labs and Taconic Biosciences, when piloting a novel RA model. In this model, animals are immunized with the glucose-6-phosphate isomerase, or GPI antigen, and arthritis and pus swelling were evaluated. The results are quite surprising. Disease development was observed in a GPI dose-dependent manner in animals sourced from Jackson Labs. In mice from Jackson, a 75% disease incidence was observed for mice immunized with 400 micrograms of GPI antigen, and a 25% disease incidence was observed for mice immunized with 250 micrograms of GPI. Similarly, mice immunized with the high dose of GPI demonstrated earlier and more severe development of RA scores than those immunized with low-dose GPI. However, mice that were sourced from Taconic, regardless of GPI antigen dose, failed to develop any disease at all. This was startling because if we had tried this novel model in only Taconic mice, we would have thought that it didn't work and that the paper was not reproducible. So just to summarize, I showed you that there were vendor differences in disease development in the GPI RA model. Disease development was observed in a dose-dependent manner only in Jackson mice, and we failed to see any disease development at all in Taconic mice. However, there are some unanswered questions from this study. We don't know if the differences in disease development were due to microbiome differences or to genetic differences from the strains from different vendors. In future experiments, we hope to address these questions by doing some co-housing or FMT experiments where we would give a Taconic FMT to Jackson mice or vice versa, and also to do some sequencing experiments in the hopes of identifying certain bacterial compositions that are leading to these disease phenotypes. I'm now going to transition it into a vendor comparison experiment that we did with our graft versus host disease model, but I'm going to start with some background on GVHD. Patients that are undergoing allogeneic hematopoietic stem cell transplant are at risk of developing GVHD, and there is an 80% incidence of GVHD at 12 to 14 days post-transplant. This is called acute GVHD. However, 30 to 70% of patients will go on to develop chronic GVHD, and this is a leading cause of death in allogeneic hematopoietic stem cell transplant survivors. GVHD involves multiple organs, including the skin, mucosa, GI tract, liver, and lungs, and is dependent on antigen-specific T cells that originate in the graft. It's also important to note that the host receiving this transplant is immunocompromised. This is required for engraftment, and it potentiates donor T cell activation. However, it can also impact the microbiome of the patient prior to this allogeneic hematopoietic stem cell transplant. Data from both the literature and the clinic support a role for the microbiome in GVHD. Patients with a less diverse intestinal microbiome have a decreased overall survival post-transplant, as well as an increased risk for transplant-related death. The converse is true for patients with a more diverse intestinal microbiome. These patients demonstrate an increased overall survival post-transplant and a decreased risk of transplant-related death. We utilized the vendor comparison strategy in our humanized GVHD model in which immune-compromised mice received a transplant of human PBMC. We compared GVHD development following human PBMC transplant in NSG mice sourced from Jackson Labs or in NOG mice sourced from Biomodels. Both of these strains are NOD-SCID mice. The NSG mouse has an IL-2 receptor gamma null mutation, and the NOG mouse is simply IL-2 receptor gamma null. The immunocompromised phenotype of these two strains should therefore be identical. GVHD progression was evaluated in these mice by daily scoring of GVHD. The GVHD score incorporates body weight loss, activity, posture, fur texture, and skin integrity, and the maximum score an animal can receive is a 10. We found that NOG mice from Taconic developed more severe disease with earlier kinetics, as demonstrated by both increased body weight loss as well as increased GVHD score in the NSG mice, as compared to the NSG mice that were sourced from Jackson Labs. This increase in body weight loss and GVHD score observed in the NOG mice was accompanied by significantly increased mortality as compared to the NSG mice. We believe that the increased GVHD observed in the NOG mice was at least in part mediated by increased human T-cell engraftment in these mice. By day 14 post-transplant, NOG mice from Taconic demonstrated increased peripheral blood levels of CD45 positive leukocytes and both CD4 positive and CD8 positive T cells as compared to NSG mice from Jackson. So to summarize these experiments, I showed you that there were differences in body weight change, GVHD score, mortality, and human lymphocyte engraftment between NSG and NOG mice. However, we have the same unanswered questions as we did for the RA experiment. We don't yet know if these differences in phenotype were due to the microbiome differences or due to the genetic differences. And we'd like to perform the same future experiments for this experiment as we would like to for the RA experiment. And in fact, we are currently performing an FMT experiment as we speak. I'd next like to discuss the assessment of the potential role for the microbiome in a metabolic study. Increasing evidence suggests the relationship between diet, nutritional status, the immune system, and the human microbiome. The nutritional status of food is influenced partially by the structure of a consumer's gut microbiota, and that food, in turn, can act to shape the microbiota itself. In this study, we utilized the antibiotic pre-treatment with or without an FMT strategy. Animals were pre-treated with either an antibiotic regimen consisting of clindamycin, ampicillin, metronidazole, or vancomycin, and vancomycin, or with vehicle for 5 days. Following a washout period, animals were then given either a fecal microbial transplant sourced from naive mice or vehicle. Body weight gain was observed for 21 days, and body composition was then analyzed by dual energy X-ray isoptometry, or DEXA. Animals within groups were then randomized by body weight into subgroups, which were fed either a high-fat or controlled diet. Body weight changes were observed for an additional 6 weeks. We found that animals pre-treated with the antibiotic pre-treatment, regardless of the FMT treatment, demonstrated significantly increased body weight gain as compared to vehicle-treated animals over the 21-day period. This increased body weight was accompanied by significantly decreased bone mineral density, bone mineral content, and bone area as compared to vehicle-treated animals. We also observed significantly decreased lean mass in the antibiotic pre-treated animals that received the FMT. Antibiotic pre-treated animals, regardless of FMT, had increases in fat mass and body fat percentage at 21 days, though these increases did not reach statistical significance. All subgroups that were fed the high-fat diet had significantly increased body weight gain over the following 6 weeks as compared to their respective control diet subgroups. However, the antibiotic pre-treated animals that were dosed with the FMT and fed the high-fat diet gained significantly more weight over these 6 weeks than did the vehicle-treated mice fed the high-fat diet. So I've just shown you that we saw differences in body weight gain, bone and fatty tissue parameters between naive mice and mice that were dosed with antibiotic with or without murine FMT. We also saw differences in body weight gain between naive mice and mice dosed with antibiotic with the FMT when fed a high-fat diet. However, we don't know how much the microbiome was depleted by the antibiotic regimen, nor do we know what the reconstitution efficacy was in animals that received the FMT. Future directions include performing sequencing on fecal pellets that were collected throughout the study so we can look at the differences in the microbiomes between the groups, and also to perform this experiment comparing weight gain between conventional mice, germ-free mice, and germ-free mice that have received an FMT. I'm next going to discuss some studies looking at the role of the microbiome in our DSS colitis model of inflammatory bowel disease, or IBD. IBD is a spectrum of chronic gastrointestinal disorders, including ulcerative colitis and Crohn's disease, and it's characterized by alternating periods of relapses and remissions. IBD is clinically characterized by diarrhea, abdominal pain, and bleeding, and is typically diagnosed by endoscopy. So IBD is usually of an unknown etiology. Both the environment, including the microbiota, and genetics are thought to play a role. We started with a simple vendor comparison experiment. Animals sourced from either Charles River Labs or Taconic Biosciences were on 3% DSS from days 0 to 5, and colitis severity was monitored by endoscopy on days 7, 10, 14, and 21. So the kinetics of weight loss were similar when comparing the two strains. The magnitude differed. The Charles River mice demonstrated increased weight loss as compared to that observed in Taconic animals. Both the kinetics and magnitude of colitis severity score, as determined by endoscopy, differed when comparing the two groups. Charles River mice demonstrated an increased colitis severity score at an earlier time point, day 10, than did Taconic mice. The peak colitis severity score in Taconic mice was lower than that observed in Charles River mice and was observed at a later time point, day 14. Based on the vendor comparison experiment, we suspected that manipulation of the microbiome could similarly yield differences in the DSS colitis phenotype. To assess this, we pre-treated mice with an antibiotic mixture consisting of clindamycin, ampicillin, metronidazole, and vancomycin for 5 days. And following a 5-day washout period, animals were treated with 3% DSS for 5 days, followed by colitis severity assessment by endoscopy on days 14 and 21. Microbiome manipulation through antibiotic pretreatment resulted in a differential disease phenotype as compared to non-pretreated animals. Mice pretreated with the antibiotic mixture demonstrated approximately 50% mortality as compared to 100% survival in the non-pretreated animals. This mortality was accompanied by earlier body weight loss kinetics. Finally, in order to assess what effect a lack of a microbiome would have on the DSS colitis phenotype, we utilized germ-free mice. These mice were treated with a lower dose of DSS, 2.5%, for 5 days, with colitis severity assessed by endoscopy on day 10. Since the animals were removed from the germ-free isolator in order to perform the endoscopy was an endpoint analysis for this study. The germ-free mice were even more sensitive to DSS-induced colitis. We observed a 75% mortality in the germ-free mice treated with DSS. This was increased as compared to the antibiotic-free treatment experiment. Mortality in those animals was around 50%, even though the germ-free mice were treated with a lower DSS dose. Body weight loss began around day 6 in the germ-free mice, and a colitis severity score of approximately 2.5 was observed. In contrast, conventional Taconic mice had a peak colitis severity score of approximately a 2. So to summarize our colitis experiments, I showed you that animals sourced from different vendors, in this case Charles River versus Taconic, responded differentially to DSS-induced colitis, both as measured by weight loss and endoscopy score. Antibiotic-treated conventional Charles River animals demonstrated increased mortality in the DSS model as compared to non-antibiotic-treated animals. And germ-free animals that were sourced from Taconic demonstrated increased sensitivity to DSS-induced colitis as compared to conventional Taconic mice, as measured by survival, weight loss, and endoscopy score. We again do not know if all of these differences were driven by the microbiota versus gene differences, and we'd like to perform follow-up experiments in germ-free mice looking at defined microbiota composition. With that, Ben is going to take over for a time to discuss the role of the microbiome in oncology studies.
Thanks very much, Caitlin. So you've just heard quite a bit about a whole bunch of different disease states and and how the microbiome and the composition of the microbiota plays a role in the disease state, and how we can model the disease with microbiome components here at Biomodels. And you've seen from the early slides a bunch of internal data that we've had the opportunity to generate just in the year and a half that we've been running our microbiome facility. In addition to those diseases, pretty much anything that has an immune component is going to be affected by the status and composition of the microbiome, both on systemic and local levels. And this provides also a nice new and novel avenues for intervention for a lot of these diseases. And this slide is just showing some of the strategies for intervention in cancer that can be utilized. So in cancer, we usually think about one of the hallmarks of cancer, which is escape from immune surveillance. And we know that the microbiota, with its role in priming the innate immune system, which then can directly modulate the adaptive immune system, can play a big role in suppressing immune surveillance of cancer and allowing cancer to carcinogenesis to happen, or cancer progression, controlling the growth of primary tumors and perhaps even in metastasis. And then importantly in treatment, especially for immunomodulatory therapies that rely on immune activities for their function. We've had a lot of great studies in the literature, and also we've seen the same internally, where the therapies have more or less efficacy depending on the composition of the microbiota. And this is important, in fact, as the targeted immunotherapies have exploded into the clinical situation. We're getting responses from some of these that are sort of unheard of, really durable responses, but only in a portion of the patients. And now there's a great deal of effort being made to sort of bring in the rest of the patients that are not responding and ask why they're not responding. And the answer may lie in the microbiome composition, and also innate and myeloid components that are either suppressing or activating the effector components that can take out the tumor. So I'll get a little bit more into that a bit later, but this first slide is just showing strategies that might be utilized to intervene in a cancer situation. Probably the earliest and the first to be adopted is just dosing of specific bacterial components. So that's using bugs as drugs, as Caitlin alluded to. Another is to drug the bugs, basically. So you use a small molecule or some other intervention to actually, or an antibiotic to change the component, the microbiome component, and then increase your efficacy. There's also many more strategies where we can use engineering of T cells to select for T cells that will respond to tumor antigens or tumor/bacterial antigens, and be able to be more effective in taking out tumor cells. We can look to vaccines, whereby we're going to use antigens that are bacterial in nature or work along with tumor antigens. So they're either oncomimetics or will be recognized in conjunction with tumor antigens to do the same thing, to elicit an anti-tumor response. Along with a number of other strategies, we can look to agents that are secreted by the microbiota themselves, et cetera, engineering of bacteria themselves. So often in our work here at Biomodels, we do a lot of immuno-oncology work. We often work in murine models and genetic models that have an intact immune system and look for efficacy of whatever the treatment is going to be. And we're often asked in that capacity to dose our animals with a standard of care as a benchmark or in combination, and we usually dose that as in a suboptimal dose so as to maximize the window where we can either improve with combination or benchmark above the current standard of care. And we do try to make all of these as clinically relevant as possible in terms of dose and schedule, et cetera. So we wanted to see how the microbiota composition might affect some of these immunomodulatory anti-cancer treatment paradigms. So basically what we did was we did a subcutaneous murine melanoma model. In this case, it's the B16F10 SIY model. The SIY is a synthetic peptide which makes the B16F10 line more immunogenic, more responsive to immunomodulatory therapies, and also increases the length of the study. It's more controlled by the immune system, so it's a bit slower. And we just ran these in A on the top. You can see it's the germ-free mice. Or in B, we have the SPF mice. So just to remind you again, the germ-free mice come in with 0 colonized microbiota, and they leave with also 0. They have nothing, no bugs. In B, the mice are coming in with whatever microbiome they've come with from their vendor, and that will be preserved. They're not going to get anything new from biomodels. They're in complete isolation. Everything is done in a sterilized hood. And we can test this by fecal sampling over time. So basically, we just gave them identical tumors and then treated here with anti-PD-L1 antibody or isotype control, everything completely identical at a suboptimal dose. And we saw some really dramatic differences in the way the tumors grew. And this is just some of the literature showing a similar experiment where efficacy of the anti-PD-L1 was different between mice sourced from different vendors, ostensibly due to microbiome differences. And they showed this through co-housing rescue experiments. So in our case, when we have the germ-free animals, they have no microbiome, and for this reason, their immune system has not been educated at all. You're starting from a different place whereby from the bottom up, the immune system hasn't seen these antigens, which are going to provide tolerance, which is going to cross-react with, not cross-react, but play on the anti-tumor, the innate anti-tumor response. Or we have the SPF mice that have a complete microbiome where the animals have an educated immune system, but you may be starting at a different status depending on what that microbiome is and how it's developed in its own right in the mouse. So you can see here that when we run the identical tumor model and treatment model in germ-free mice versus mice with an intact microbiome, the tumors grow slower overall. But when we treat with the anti-PD-L1, we don't see a response in the germ-free mice, while we are able to see a response in mice with a microbiome. And why that is sort of beyond the scope of what we did here. And the mechanistic insights are coming with the literature. We learn more every day, but there's not a really great understanding of the pathways involved. In some of the cell types of interest, of course, we always think about the effector cells, but the microbiome as it evolves over time with the innate immune system, it's some of our earliest immune system evolved to control and to allow the beneficial microbiota to persist. And this is, the immune system is probably the innate immune system is probably the first line for the activities of the microbiome in terms of eliciting tolerance or activation. So we look at dendritic cells, for example, or other antigen-presenting cells, myeloid cells, and innate lymphoid cells. And these cells are definitely of interest in terms of the mechanistic underpinnings of the microbiome in many of these disease states. So this little figure that I've put up here is just showing differences in local and systemic PD-L1 expression of pulmonary CD11b positive pulmonary dendritic cells. In this case, you have the SPF mice with a microbiome are now upregulating PD-L1 in the lung versus the germ-free mice. And while systemically, the splenic DCs are really don't show any difference. Both high there. So back to the data. So if we looked at, when we looked at the mice with differing microbiomes, both have a microbiome, but they're different. The taconic mice are, this is the same data from the last graph, just overlaid with the other comparison now. So the taconic mice do respond to the PD-L1. And in this case, the Jackson mice did not respond to the suboptimal dose nearly at all. And so, again, as Caitlin alluded to, if you ran this in Jackson mice alone, you would think, okay, maybe it's not working, something is not right. But basically, what you can see is the Jackson mice are starting at sort of the same place as the anti-PD-L1 treated taconic mice. So really, what we're looking at are different starting points. And so when you're running treatments, testing potential treatments in these models, you need to consider where you're starting and where you're ending, not only where you're ending and just assume you're starting at the same place. Now, if we were to provide a larger, a more concentrated dose or up the schedule, we would see response from the anti-PD-L1 in the Jackson mice as well. It's just a matter of knowing, just keeping it in mind and considering it. So not only for treatment paradigms that are relying directly on microbiome modulation, but also any immunomodulatory therapy, the microbiome should really be considered as a key component in interpreting that data. Also, as Caitlin pointed out, we have the capability here at Biomodels to go quite deep and to look into the immunological components of the treatment, where we start and where we end after therapy. And in this case, we're looking at tumor-infiltrating effector T cells. We looked at CD4 and CD8, also interferon gamma, in this case by qPCR. And as you can maybe see from this kind of busy slide, we didn't see a whole lot of differences. We saw we did see some real differences in the baseline, but not so much in the when we compared the treated group to the non-treated group, the effector. And this may be a product of the fact that the effector cells are not really the ones being directly affected by microbiome, but rather their activation or suppression is really mediated by microbiome via the innate cells or the myeloid cells. So just to sum up, for this particular study, we saw differential tumor growth in response to treatment to this anti-PD-L1 in animals with differing microbiomes or no microbiome at all. And this is just one way we can do oncology using germ-free gnotobiotic mice, using microbiome modulatory treatment paradigms and looking at the effects on the immune system for immunomodulatory anti-cancer therapies. So thank you very much. I'm going to throw it back to Caitlin now.
All right. So just in conclusion, we've talked about how the microbiome can play significant roles in human health and disease, as well as in the response to therapeutic interventions. And we've gone over that Biomodels offers a number of experimental approaches for probing disease models with a microbiome component. Finally, we went over several examples of murine models and how we looked at the role of the microbiome in those models. In a novel rheumatoid arthritis model, we showed differential disease development in animals sourced from different vendors, in this case Jackson Labs versus Taconic. In a humanized graft versus host disease model, we showed differential disease development and T-cell engraftment in animals sourced from different vendors, in this case NSG mice from Jackson versus NOG mice from Taconic. In the metabolism study, we showed that there was increased body weight gain in antibiotic-treated mice versus vehicle-treated mice. And in several colitis studies, we showed differential disease development in animals sourced from different vendors, in antibiotic pre-treated animals, and in germ-free mice. Then finally, we showed that there was differential tumor growth and cancer immunotherapy responses in animals sourced from different vendors, in this case Jackson versus Taconic, and in germ-free mice from Taconic. So we'd just like to take some time to acknowledge all of the people that made this work happen, especially our Chief Scientific Officer, Steve Sonis, and our Chief Operating Officer, Greg Lang, as well as our fellow scientists, business administration, and technical staff. Thank you, everyone, for taking the time to listen to our webinar today, and we'd be happy to address any questions by email, and thank you again for your time.
Thank you very much, Caitlin and Ben. We'd like to thank Biomodels for sponsoring today's web symposium, and we'd like to thank you all for attending today. We really appreciate it. Thank you, and have a great day.